Thanks, everyone, and thank you to the AMI Online Learning Committee for inviting me to speak with you today. Before I begin, here are my disclosures. And I'm just going to dive right into, there's a lot to cover today, and what I've learned is but a tiny aerosolized particle of information, but I hope to give you all top-level understanding of the novel coronavirus and COVID-19 from a medical illustrator's perspective. So I want to start with this thought. This whole pandemic had me thinking about the world's response to 9/11. I tell a lot of people about this. I was at work that day, passively listening to the radio, and I sat focused with tunnel vision on impending deadlines. And once I realized what was happening in the news, none of that mattered. People were dying. My role as a medical illustrator didn't matter. My clients weren't going to care about deadlines. But I felt utterly useless. With COVID, we face a similar predicament. People, again, are dying. But research marches on, and our field has seen a surge where we couldn't illustrate fast enough. We as a community have the power to use rapid, widespread, effective communication of trusted factual information to lift science. So unlike 9/11, I feel needed. We are needed. That goes without saying. Just last week, the Beasley Design Awards recognized Alyssa Eckert and Dan Higgins for the most widely seen visualization of the virus, and it was named the 2020 top graphic. With our response to the coronavirus pandemic, associations now name the AMI as one of the top 100 associations that will save the world. So we have a responsibility. Back in February on the AMI Hub, Michael Konemus proposed that we start a virtual group since things seem to be transitioning into a dangerous pandemic, and we as medical illustrators were in a very unique position to be able to share the latest accurate information with the public. So with that, the COVID-19 Medical Illustrators Group was formed on the AMI Hub, and we began compiling what is now an 89-page resource by 24 contributors called the SARS-CoV-2 COVID-19 Research for the Medical Illustrators Google Doc. And anyone who wants to see it is free to see it if you go on the Hub. Though it's impossible to say this document is or ever will be complete, there you will find references, resources, commentary, and visualizations covering a wide variety of topics focused on the novel coronavirus. This is the handbook you all should know about. Many continue to refer and contribute to it for every COVID-related project. So be sure to thank Michael, because this has really been an invaluable asset to our association. So remember back in March? This is where the journey began. Alyssa and Dan put a face to a name. I asked them to share their thoughts on this invaluable contribution to public health education. These two artists explained that creating visual representations of diseases provides a way to take something complex and abstract and make it tangible through visualization. One goal is that through this visualization, we can help inform and educate the public about the disease, and thus, hopefully, help improve public health by limiting and halting the spread of the disease. And the CDC has played an invaluable and an integral part in imparting this message into our everyday lives. I see this sign everywhere I go shopping. It's great. So back in mid-March is where my story began. Every time something like this pops up, the Bloomberg School of Public Health is always there, and sure enough, there's a knock on my door. They are planning the next issue of the Hopkins Bloomberg Public Health magazine and asked for my help. This would be a very open-ended infographic for the COVID special summer issue, and they had already tapped a world-renowned Bloomberg virologist as a content expert. So of course, I logged into the AMI Hub, downloaded the Google Doc, I pored over all the resources listed in preparation for my meeting. And when I met with the editor and the art director, I wasn't given a story to tell. I had to learn everything I could and begin to interpret what I was seeing into my own visual narrative, and eventually the illustration would write its own story. So soon after, the team and I met with Andy Peacock. I was introduced to the co-director of the Johns Hopkins Center of Excellence in Influenza Research and Surveillance and professor of molecular microbiology and immunology. He's the guy that's at the front of the room and is in the center of the table. He's guy number 3, who's kind of looking to the side. His voice would narrate that story. The day before, as you can see in this picture, this is March 6th. So notice, nobody's wearing masks yet. And you see the COVID dashboard up on the wall in between the paintings of different politicians, I'm assuming. The day before, he and several Hopkins University experts in public health offered a briefing on Capitol Hill for officials seeking facts and perspective on COVID-19. Congressional staff and media heard directly from Andy and his fellow researchers, Hopkins experts who have been at the forefront of the international response to COVID-19. And they were advising on best ways to mobilize resources, improve care, and save lives. So, of course, I had way too many questions swarming in my head. What are we drawing that hasn't been visualized by the CDC already? And what will still be relevant by the time the summer issue comes out? Andy began with the most important facts from structure to life cycle, viral genome, and what proteins were encoded within, facts about its epidemiology, potential targeted therapies, and of course, what mysteries still needed to be uncovered. This would be our last in-person meeting for this project because on I'll never forget March 17th, that was our last day of being in person. So teaching and creating visualizations would continue at home. So let's talk about viruses for a moment. To understand coronaviruses enough to build visuals about it, we have to start with some basics. Viruses come in all shapes and sizes. No matter the form, all viruses have 2 things in common. Genetic material, DNA or RNA, which encode for its own protein makeup, and a protective protein coat called the nucleocapsid. So from there, we have 2 types of viruses, enveloped and non-enveloped viruses. And you can see the long laundry list to the left and right of each of these. The envelope is simply a layer of lipids that surrounds the capsid, and in many cases also forms a mosaic of other proteins. That's enough for me to explain to my kids, but I wanted to start developing a more mature visual language that could be used to explain what this virus looks like in an oversimplified style before diving into a more 3-dimensional depiction. So I'm just showing you here kind of what is this virus in a nutshell? What is the essence of the virus? What are the physical structural proteins that you will find? Spike, membrane or matrix proteins, the envelope protein, the nucleocapsid proteins, and then the RNA and the lipid layer. And then also, I wanted to thank Jane Whitney for allowing me to share her beautiful and very elegant depiction of this simplification of the virus. And then you can also see how it binds to the ACE2 receptor there. So now let's just kind of look at its phylogeny for a moment. Coronaviruses are a large group of enveloped single-stranded positive sense RNA viruses that infect a wide range of avian and mammalian species, including humans, of course. And for people to really understand its complexity without being too complex, let's just kind of distill this down. The most well-known and common human coronaviruses from recent outbreaks include SARS-1 and SARS-2, also known as severe acute respiratory syndrome coronaviruses, and then, of course, the Middle East respiratory syndrome coronavirus, or MERS-CoV. Here's where my conversations with Andy Peacock began. This particular virus, there's no known host species from which it has mutated, although likely researchers are saying bats or pangolins. And if you look at that phylogenetic tree, it actually makes sense because you see bats surrounding it a lot. So I don't know, that's just a speculation, but there are there's plenty of research studies that can tell you what it can infect. And that includes bats, cats, dogs, tigers, lions, minks, monkeys. There's a lot. And there's also like what does it not infect? Poultry, pigs, no. Okay. So when the virus binds to the lung's ciliated epithelial cell surface via angiotensin converting enzyme 2, or ACE2, it does so with great affinity in humans. Recent lab studies on CoV-2 variants suggest mutations in the spike protein improve this even more. And a protease, that's the yellow protein that you see there called TMPRSS2, cleaves specific regions of the spike, which allows the virus to enter the cell. Yes, it's dreadful, but Andy also shared some good news. So SARS-CoV-2 has a very low, slow mutation rate. A non-structural protein, or an NSP2, acts as a proofreader. And in lab experiments, if this protein is knocked out of its genome, the virus, being less tolerant to the host's immune response, will mutate itself to death. In fact, the virus doesn't want to mutate if it doesn't have to. It doesn't like it. It wants to avoid it. Because mutations have a slow enough prevalence in SARS-CoV-2, it's easy for researchers to figure out the genetic epidemiology, making its spread easy to track within transmission chains. So, for example, we know that it went from China to Iran, and then from Iran to Italy, and then from Italy to Boston, Massachusetts. We can figure those things out just by looking at those tiny little mutations that occur at a slow rate. So I began to pore over the literature before me and, of course, researching the molecular structure of the virus. And I often, when I'm working on something like this, I often will take illustration notes. So I'll create these files, like these libraries. You can use Adobe to create your own asset libraries that I can refer back to that has the articles or models that I was exploring. Many of the novel coronavirus proteins were available in the PDB, but earlier last year we were only seeing a handful that were updated from SARS-CoV-1. Others trickled in and continue to be updated as they become known, and of course I relied on the research that was being documented in our shared Google Doc. But also, I began collaborating and conversing with others, searching for the same answers, and talk about being, socially isolated. I mean, this is that connection that you can get from the AMI. We were correcting things together. We were adding our own findings. This led to some really fascinating conversations, either via messages on social media, through emails. I had a few Zoom calls. It was really great. When we're looking at the host cell proteins, I just want to point out a few things. So here you're seeing ACE2. And if you're ever in doubt, like, what does it look like? It's just two boxing gloves. It's a dimer. And then the other protein that you see in yellow, that's TMPRSS2. But if you ever go into the PDB and you're downloading some of these proteins, you might be confused when you see extra structures there. So I don't want anyone to be confused. I'm going to just point out on the right-hand side, you'll see above ACE2 some purple and tan connections there. That is the receptor binding domain of the spike protein. So that's part of the spike protein that's flipped out and binding to ACE. So that's what it looks like when it binds. And then laterally, you see another purple and a green protein. That's B0AT1 or BOAT. And those are found in the intestines. Now, I don't think that they're found in the lungs. So if you're depicting this in the lungs, you don't necessarily have to show BOAT. So I'm glad we ' that out of the way. Okay. When we're looking at the virus's structure, the coronavirus positive sense RNA genome has a 5' methylated cap and a 3' polyadenylated tail, which allows it to act like messenger RNA. And it can be directly translated by the host cell's ribosomes. It basically takes over the host cell's machinery. The host ribosomes translate the initial open reading frames, or the ORFs that you see up there at the top, and undergo protease activity where these polypeptides get cleaved at different specific sites. This eventually yields 16 non-structural proteins. You'll see the name NSP 1 through 16, and they also have their own individual names. Products include various replication proteins such as RNA-dependent RNA polymerase, which is known by many different names. RDRP, RNA replicase, NSP12. So if you ever run into that, don't be confused. It's the same thing. There are very specific genes that will undergo transcription and translation, as I mentioned, using the host's own replication machinery in order to physically assemble structural proteins and release new virions. And in the case of coronaviruses, this includes this crown-like spike trimer, the membrane or matrix dimer, and the envelope pentamer. The nucleocapsid surrounds the RNA in a helical fashion, and each of these proteins has specific structure and role in viral entry, replication, and assembly. And here you can also see where various microbicides can act to disrupt the transmission from surfaces and from your hands. So of course, if you destroy that envelope, the virus can't get into your cells. So the spike protein is perhaps the most visualized of all the coronavirus proteins. Dr. Rami Amaro contributed to everybody's understanding of the spike protein's highly glycosylated structure through another webinar she gave earlier last year. I highly recommend that you watch that if you haven't. The spikes act as transmembrane keys that unlock the host cell to infection once it binds with the host cell membrane. And the S1 residue interacts via its receptor binding domain, so you can see that right there, and the ACE2 on the cell surface. And that's what initiates infection. So RBD is also the target of many of the vaccines you're hearing about. So you'll see antibodies produced during the host's immune response will bind and therefore block the spike protein from initiating cell entry. There's a beautiful illustration done by Veronica Falconeri-Hayes, and she created this visual that shows this great detail of how the spike trimer has two subunits. S1, which acts like a cap, which includes 3 RBDs that can act as a hook and unlock the chain of events that lead to cleavage and the removal of the cap and all that stuff. And then S2 that acts like a tether once the protein refolds and pulls the membranes closer together, leading to membrane fusion. David Goodsell shared some great insight and pointed to the newest research having to do with the spikes, the flexibility of the spike. So those spikes do move. The other thing I wanted to point out is that recently more and more literature is talking about the evidence that the spike protein has specific regions of flexibility. So we're talking about hinge points, like specific hinge points, like a heel and an ankle and a knee. And in the background here, you can see one of Val Altunian's beautiful depictions of a spike protein kind of leaning down. I'm sorry that the article's covering it up. I apologize. So this protein has 3 conformational states. This is a beautiful graphic by Gloria Fuentes, who had worked with Gail McGill. So here you see the pre-fusion closed state, pre-hairpin intermediate state, and post-fusion hairpin state. During membrane fusion, a trimer of hairpins forms. I also had a recent conversation with Gail McGill, and it was it was one of these fascinating Zoom conversations about the flexibility and the conformational changes that occur before and during membrane fusion, and the way the 2 layers fuse, and this spike. We were talking about all kinds of stuff, spike protein mutations. Thank you so much, Gail. I so appreciate your insight and your knowledge. Take a look at the Digizyme team's animation in the 2020 AMI Salon because it shows this part of the story so clearly. I hadn't seen this idea until Gloria shared some models with me, and then Gail explained this whole process to me. Gail allowed me to share his visuals, and I wanted to use this short clip, so I'm going to kind of walk you through it. Here you see the spike protein hanging down from the virus. And what's going to happen is you see there's the S1 cap, the S1 domain on the bottom, and the S2. And what happens is RBD flips out, and it's going to bind to ACE2. And once that occurs, the cap is released, and now the spike is exposed, and it will tether itself to the host cell that you see down there in blue. And now the protein begins to refold itself, and this action is what's going to pull those two membranes together and fuse. Thank you so much, Gail. So this is so cool. I was watching David Goodsell. We had a few email conversations, but then also, on social media, he's been posting his progress of his paintings, and you can see this more recent depiction of the SARS-CoV-2, and it's this very moment that he's depicting, and you can see some of those hairpins, and then you can see a spike trimer that hasn't been, the cap hasn't come off. These viruses have both of these conformations, so it's important to kind of note that. The envelope protein, this is an enigma, and it's very interesting. So this is an ion channel. I call it my cute little pink fluffy mushroom cloud. It's a protein whose structure is still being studied, and because so much of the data we had last year was from SARS-1, for the most part acts as a viroporin. It self-assembles in host membranes, so the Golgi is what we're talking about, forming what is likely a pentameric protein lipid pore that allows ion transport and plays a central role in virus morphogenesis, assembly, budding, envelope formation, and the induction of pathogenic apoptosis. Even though a high number of these proteins are made inside the host cell, only a small portion is incorporated into the virion envelope, and far, far less in proportion to S, N, or M. Here's something that had me second guessing myself. Most depictions you will see have this mushroom cap facing outside the virus. So I had, again, just last week, I had a very interesting conversation with Alex Nazlidis, and Evelyn Meisels pointed this out to me. And Alex is a UIC grad student who's been working with Evelyn, and he found in his research through simulation and new models that are out there that we are probably placing our E proteins in our SARS-CoV-2 models incorrectly. It's a question I've been second guessing in my own work. He sent me this great new model. It's up there in Uniprot, and also the transmembrane portion of for SARS-2 is now listed in the PDB as of 2020. But here's what he's finding. So if you look at the slide, N-terminus extravirion, C-terminus intravirion. What does this really mean? So here is what Alex is suggesting. So the C-terminus, so on the left you're seeing the SARS-CoV PDB entry, which was posted back in 2017. And the C-terminus is facing outward. So you can see the spike protein for reference outside the virus and the N-terminus inside. But if you look at the new model, based on this PDB, the transmembrane portion that you see in the inset, that's just the mushroom stalk. The N-terminus is actually facing outwards and the C-terminus inwards, and the mushroom cap is at the C-terminus. So this is very interesting. I'm scratching my head and I'm looking at my illustration, and so I went back to another article and I was rereading it, even though it's from last year, before this data came out, and the literature actually talks about this enigma. When I go back and I look at what we do know, that it's most prevalent in the host cell, not necessarily the virus, perhaps this protein does in fact sit this way for ion transport when it's sitting in the endoplasmic reticulum or Golgi. So you can see here, I actually am highlighting just a little vesicle of the Golgi, and then I've blown up and kind of diagrammatically shown what I'm talking about. What happens is these proteins, when they're produced in the host cell, they're getting packaged in the Golgi, and you know that the Golgi then begins to expand and wrap around, and then it packages a piece of RNA in there. So it goes inside out again. So yeah, it's fascinating. if this is true, then here's what Alex is suggesting. We all need to flip our E proteins. A man can work from sun to sun, but a medical illustration is never done. And it's true. I mean, it is just not ever done. And Melanie could tell you all about it because it's just never done, right? So I don't know if you've ever seen, again, go to the salon. You'll see Melanie Connolly's beautiful depiction of how a medical illustration can mutate. So, when we don't have enough information, we're kind of guessing, and then Rami Amaro spoke to us, and then this happened, and then this happened, it's just really cool to see this. All right, a few more proteins, and then we're going to move on. The matrix, or membrane protein, is the most abundant protein in the viral envelope, and it plays a central role in the virus morphogenesis and assembly via its interactions with other viral proteins. That's the purple protein you see there. It's also, for the medical illustrator, the most mysterious to capture visually. Only remote homologs were identified as potential template structures. So because the M protein is the most abundant structural protein in the virus, it defines the shape of the viral envelope and acts as a central organizer in viral assembly. It does interact with the S, the N, and the E proteins. And then the nucleocapsid that you see down there in kind of baby blue is similar in structural topology to the Ebola virus. The nucleocapsid protein packages the positive strand viral genome RNA into a helical ribonucleocapsid and plays a fundamental role during virion assembly through its interactions with the viral genome and the membrane protein. So there's also 16 non-structural proteins, as I mentioned earlier, but I'm not going to go through all of them. But their roles play out in the playground that is the host cell. And it takes, as it releases the RNA, it takes over the cell and begins to translate it. We're not going to talk about all of these, but I do want to mention just a few. So on the left, you'll see the green one, NSP2, as mentioned earlier. This is that protein that Andy was talking about as a modulator of the host cell survival signaling pathway by interacting with proteins to check with viral genomic mutations and maintain the functional integrity of the mitochondria, therefore protecting cells from various stresses. So if you pluck this out of the genome, it's going to mutate itself to death. NSP5, which is the orange one, if you take your hands and kind of place them in like a heart shape, like put one palm facing your face and another one like kind of like mittens, you'll see it kind of crosses over and it looks like heart shape, but that's actually two proteins that I colored in orange. This one cuts the viral polyproteins into functional pieces, and you can see that little green thing in there. That's actually can act as an inhibitor. We'll talk about that later. And then NSP12, again, that's RDRP. So how are all of these structures put together? Let's just quickly look at some geometry. These are approximate numbers, except for the genome, which is 29,903 nucleotides to be exact. But these numbers, it depends on which paper you're reading and which research study you're looking at. For the most part, the virus particle is no more than 100 nanometers in diameter. It's spherical or ellipsoidal. The N dimer can accommodate an 8M dimer to 4N to 1 spike trimer ratio. Earlier on, 74 spikes per average size coronavirus, that's what I was seeing, but recent studies suggest it's far less, 20 to 40. There are pre- and post-fusion spikes that are present, and their ratio changes as new variants come about. There's about 1,100 M dimer proteins per virion, and the N protein can accommodate between 14 and 40 nucleotides. So we're just going to look at a few depictions now, a few visualizations of the SARS-CoV-2 by some of our colleagues. I told Nick Klein that I thought his depiction was so beautiful. I'm I'm telling you, Isoform, you need to 3D print this to a size that will fit very nicely on my shoulder as a brooch, and you need to send it to me. I think it's gorgeous. Here is another example by Nick Woolridge. I believe he has this posted on the Toronto site and also on social media, and I believe that he even animated it. And then, of course, Veronica did this amazing article for Scientific American. She illustrated this article, and so here is her version of the virus. Once this virus finds itself into our upper respiratory tract, they're transmitted from one host to another host, depending on the coronavirus species, either by aerosol, various objects and surfaces. Human coronaviruses infect the respiratory epithelium, while animal coronaviruses generally infect the epithelial cells of the digestive tract. I never ended up using this next infographic. I thought it was interesting to learn what favorite spots different viruses like to invade and how it leads to severe disease. So in this infographic, there are two very different phases of COVID-19. In phase 1, infection takes place in the upper respiratory tract, where epithelium is several layers thick. The virus targets and kills ciliated cells, leading to the early symptoms like the runny nose, itchy throat, and then as cells die, there's less mucociliary function, and therefore cell motility and the ability to move or clear mucus is inhibited. Instead, this thick fluid sinks down into the bronchi and bronchioles, and as this happens, the immune response kicks in. Now, if the immune response is not enough to help a patient recover, this will lead to an inflammatory response. Now we're headed to phase 2. The infection sinks lower into the lungs' thinner tissues where it can't get out, and it targets the type 2 alveolar cells. The damage sustained by the alveoli, of only one cell thickness, remember, gives rise to pneumonia and acute respiratory distress syndrome. This is deadly. Those who suffer from chronic diseases like lung disease, diabetes, cardiovascular disease, hypertension, or even advanced age are at highest risk for an inflammatory response called the cytokine storm. And this is an uncontrolled overproduction of soluble markers of inflammation, which in turn sustain an aberrant systemic inflammatory response. But this is, like I said, this is a life-threatening condition where patients suffer severe shortness of breath and often are unable to breathe on their own or without the support of a ventilator. So the process of creating an engaging, relevant, and audience-specific infographic began with drafting a logical, visual, and descriptive story with multiple layers of information. I realized that much of the visual materials put out by the CDC served as exemplary resources for educating the public about the novel coronavirus. So instead, our goal was to change the perspective of the everyday narrative, social distancing, wearing masks, washing your hands. We wanted to place the story within the context of the researcher's voice, giving the audience the same privilege I enjoyed in discussing strategies in the works to stop the pandemic with a real virologist. So let the audience be the interviewer is what I was thinking. What answers might a virologist be searching for, and then answer them? How is the novel coronavirus, which preys relentlessly on its victims' weaknesses, having its own weaknesses targeted? I was trying to figure out how to tell the audience what made this virus unique, so the structures that we talked about, how it replicated, and then the current research into potential targets that could lead to effective life-saving treatments. So here is where I'm showing the replication cycle. So it comes into the cell, it releases its viral RNA, it goes through this whole process of replicating itself, so the RNA, but also creating its proteins, and then repackaging in the Golgi, and then it comes out of the cell through exocytosis. The other thing that we were focusing on were potential research targets. Monoclonal antibodies and convalescent plasma. Acts very much like what a vaccine is supposed to do. Recovered patients, they have antibodies to the virus in their blood plasma, and by giving those antibodies to patients with active COVID-19, this can be effective in fighting the proliferation of the virus. Very much like the vaccine. It's not a long-term fix, but it can help. Now, okay, before you freak out and you go, oh my God, chloroquine and hydroxychloroquine, what are you talking about, Jenny? We did this on purpose. We did not want the audience to even guess what we thought about this. We didn't want to leave it up to the audience, so we put it in there, and we stated specifically that while these drugs appear to offer no significant benefits, they actually increase risk. Okay, these are drugs that are used for malaria, and it's a different you have to understand, like, the way that malaria gets into the cell. With clathrin, like, it acts on that, and that's not necessarily what we need to be focusing on. So, there was one scientific study that came out of the Henry Ford clinic, and it has flaws, and it looked promising, and then everybody thought it was the miracle drug. So we wanted to make sure that we told the audience, no, this is not, something that people should be taking. ACE2 inhibitors make sense, right? We want to inhibit the binding of the spike trimer to the ACE2 receptor. So targeting ACE2 is another target. Inhibiting protease activity. So again, that little green part that you see inside the main protease can kind of lock itself in there and block it from performing the essential functions it needs for the virus. And then also inhibiting RNA polymerase. You've probably heard about remdesivir, and this is an interesting target. It's a nucleotide prodrug therapeutic given to patients once diagnosed with COVID-19, where their predisposition to severe inflammatory responses can lead to hospitalization. So it's an FDA-approved antiviral treatment. It shortens the recovery time. And in fact, there's even studies on taking remdesivir and making it into, adding a bond between it, so that it's called flex remdesivir, and it actually can get in there and bind more tightly, and act as a dual target. So not just on RNA polymerase, but also on methyltransferase. So I thought that was really interesting. It was another project. And then, of course, finally, vaccines, the thing that we've all been waiting for. So I'm going to touch on that in a minute, but June 2020 came, and I put my pencil down, and I turned in what I had at the time, which is what you see here. So this 2-page spread was published in print and online, and before I gave it to the magazine, I put it together as a poster because I knew I was going to need it to be modular for its various publication uses. So the illustrations were purposefully constructed with 2D animation in mind. Right as the magazine hit stands in the quiet halls of the School of Public Health, we were busy working on that. So the animated version of the replication story, with this, we wanted to speak to the audience with the voice of a virologist, literally. So let's just take a pause and listen to Andy as he narrates.
What a virus wants to do is make more copies of itself and send it out. So coronaviruses are a rather large family of viruses. They are RNA viruses. Particularly in recent years, we've had viruses such as SARS-CoV and MERS, which are coronaviruses that have made the jump from another animal to humans, but have been associated with much more severe disease. And so SARS-CoV-2 represents the third of that, what I'll call the severe coronavirus diseases. Coronaviruses all have a very similar genome structure, but based on the specific sequences of their genes and the number of genes they have that are specialized for pathogenesis, you can differentiate one from the other. So the first thing a virus has to do is find a way to attach itself to a cell so that it can specifically get in there. That's the virus receptor interactions. And for SARS-CoV-2, it binds to a protein called ACE2 that's on the surface of some but not all cells. Viruses then have to get inside a cell, in the case of SARS coronavirus, it has a sort of a membrane around it that hides its genetic material from the outside world. Your cells have the same kind of membrane around them that hides their material from the outside world. So when those two things come together, the virus has to find a way to get its genes inside the host cell. The virus genome, when it enters a cell, starts to find something in the host called a ribosome. And a ribosome is what turns RNA into proteins. It takes the sequence of the RNA and then it translates that into a protein sequence. RNA is primarily the means that we have that express our genes. The more of the viral RNA that's in the cell, the more of that RNA will be translated into proteins, and the faster the virus can take over the cell. Their RNA serves those two purposes. It encodes the genes that it has to complete its life cycle, but it also is the way that it sends a message out to the cells to say, make more of my genes. And so within a few hours, there's a large portion of your proteins within your cell that are either from the virus or that the virus has somehow manipulated. And then towards the end of the virus life cycle, the virus sends out its proteins to particular places within the cell that are then the places where proteins are concentrated and new virus particles are made.
I recently wrote to Andy, as I still want to meet with him about another project that I'm working on, and of course to get his thoughts about the current vaccine and variants landscape. He's a really hard guy to pin down. For now, what I'm going to share with you is what I know from other projects that I've been a part of in the past. This data was pulled last week and shows the current state of things in terms of what's out there and what's being developed. So ultimately, vaccines generate an immune response, which is what we would see when infected with a pathogen, but without the severe disease involved. So our immune system, the cells we find in the lymph nodes, are primed and they're ready to attack the real virus with the antibodies produced by our B cells. Many of us think about the flu vaccine or other common inoculations where essentially we might be injected with an inactive virus. So you can see that it would generate this response, but the Pfizer-BioNTech and the Moderna vaccines are genetic or mRNA vaccines. So to many, this may seem like a new thing, but it's actually, in reality, biotechnology that's been in the works for decades. This illustration is one that I created nearly 20 years ago for a company that's working in the siRNA and mRNA therapeutic space. They're studying and creating drug therapies for very specific diseases, creating customized pharmaceuticals tailored to their patients. So things like amyloidosis, ALS, cancer therapy drugs, this is nothing new. And of course, vaccines are nothing new. So we know scientists have the technology to modify vaccines year after year. We see this every year with the flu vaccine. Essentially, what an mRNA vaccine can do is very similar to inactivated virus vaccines that we see. Here, what they're doing is they're basically taking just a part of the genome that codes for the spike protein. They're isolating it. They're putting those sequences into lipid-coated vesicles, or liposomes, I should say. They're cholesterol-coated so they can get into the cell. These are what the vaccines are. They're injected into the patient. The vaccine enables a protective immune response that comes from the lymph nodes. The mRNA gets into the cell and begins to be translated into, so in this case, a spike protein that gets expressed on the cell surface. This is recognized as something foreign by a T cell immune response, which then stimulates antibody production in B cells. And so then when you're infected with COVID, hopefully it works, right? So that's essentially what it does. And for me, it's exciting to see members of our association at the head of the table partnering with these companies to tell their stories. Here, Cognition Studio made the cover of Molecular Therapy at the beginning of this month, and you see a beautiful depiction of this liposome with several copies of the mRNA inside. Another good cell painting because I just love them. It's really neat because what you see here is the liposome. You see the purple mRNA. You see polyethylene glycol in green that are on the outer surface, and then you see those little triad antibodies that are on the outside. I'll just conclude with other visualizations and just some more eye candy from David Goodsell because it's just so good. And this is great. So you should know about this. At the National Institute of Allergies and Infectious Diseases, Kristen Brown had worked on a model that you can download. She's also working on a fabulous app. And on the right-hand side, you can see my 3D printed version of it. So for me, it was kind of exciting. It's scary and exciting to say, like, oh my God, I'm holding COVID in my hands. But it is kind of cool to actually have a physical model of it in your hands. And then, of course, another beautiful visualization by Cognition Studio. I'm not going to show you the animation that I created of this because it will make you queasy. But if you do want to watch it, you can. I'll share there's a YouTube link down there in the corner. We've been doing a lot of different things at Hopkins, and interestingly, at Hopkins, if you are infected with COVID and you're showing symptoms, then you can certainly get a nasal swab, but you can't get one unless you're symptomatic. But now they have this new test, the passive drool saliva self-collection test. So if you get a call from a contact tracer saying that you might have been exposed, you can immediately go and get this type of a test, which is really nice. And then if you want to see other visualizations, both on the AMI.org website, you'll see lots of different contributions by AMI members, but also go ahead and take a look at the AMI Salon. I created a COVID-19 gallery. It wasn't a separate category, but I thought it would be really nice for people to be able to just quickly see all the entries that had to do with this. So kind of looking back at where we started and where we are now, I know it's a sobering view, but what I want to impart is that I can't tell you what an invaluable experience it's been for me to be a part of this group and to learn from and connect with other AMI colleagues. We are really having a huge impact on the public's understanding of this pandemic. Thank you again to Michael for initiating our COVID group and to all of the contributors. Thank you to the AMI, the Online Learning Committee, especially Lydia, Dorothy, and Michael. And thank you all for being here and listening.